All right. Thanks to everyone who's made their way back to the day 2 of our Human Cell Atlas meetings. I hope everyone's having a good time in Cambridge. Unseasonably warm weather, but a little rainy. We've sort of been charged with the Human Cell Atlas conversation on heart and vascular. And I will say, and I'll say this probably over and over again, that we have the hardest task because we're given like 2 completely different organ systems. Other than the fact that they both come in direct contact with blood, there's really no difference, no similarities between vascular and heart, different cell types, different diseases. So I think that will be sort of our challenge today. And I want to sort of go through some of the current work that's going on in heart and vascular disease, single cell genomics. And then I want us all to sort of engage actively. I went to the immune session yesterday. I thought it was great. There was a lot of problem solving, a lot of talking about the challenges that exist with single cell analysis for the immune diseases, immune cells, standardization, best protocols. And for us, I really don't think that we're even that close to a first draft atlas yet. So that should maybe be the sort of goal at the end of this, is if we were to have a first draft human cell atlas, what would it look like? So first of all, I guess two announcements. This is being webcast, as you guys all know, on YouTube. For that reason, they were requesting that most people sit in the first 3 rows. It does look bad and not as interactive if people are all the way in the back. And then also, sort of along those lines, is that if you do speak, you will be webcast on this YouTube platform. So those are the only announcements. I wanted to introduce myself. And then before we get started, maybe the 4 of kind of like us co-moderators will also introduce ourselves. But my name's Raj Gupta. I'm a cardiologist at Brigham and Women's Hospital. I did a postdoc here at the Broad Institute with Sate Katharayson on the genomics of coronary artery disease. And one of the projects I worked on, actually sort of the focus of my postdoc, was a SNP that was associated with 5 vascular diseases. And I identified that it had a regulatory effect only in endothelial cells. So in a lot of these sort of GTEx, transcriptome-wide analyses, that association was lost because endothelial cells only make 3% of a blood vessel. And so that got me interested in the single cell architecture of a blood vessel, and that's sort of how I'm here. I've been working with the Regev lab on protocols for vascular tissue, primarily, not as much on the cardiac tissue. And then, we have 3 other disease and methodology experts here. So, Rick, do you want to just say hi to everyone?
Sure, hi. So, hi, I'm Rick Mitchell. I'm a cardiovascular pathologist, also in Boston at Brigham and Women's Hospital. My research interests are primarily in acute chronic allograft rejection, specifically for heart, but it's applicable to a variety of other solid organ transplants. I've done extensive work collaborating with Peter Libby and others at Brigham, looking at also atherosclerotic disease. So heart vessel are no strangers.
All right. Barbara?
Hi, I'm Barbara Engelhardt. I'm a professor at Princeton University. My background is in statistics and machine learning. I'm interested in sort of both the processing and the analytics of the data, but also experimental design. So I hope we get to talk about all of these aspects of it today.
Michela.
Hi, I'm Michela Noseda. I come from Imperial College in London. I'm a cardiovascular biologist. I'm interested primarily in paracrine mechanisms regulating and maintaining cardiac homeostasis and what happens after ischemic injury. I came into studying to use single cell approaches because to dissect the stromal compartment of the heart. I started with very simple single cell qRT-PCR, and then fortunately all of these new technologies came along, and I started to collaborate with Sarah, with Norbert in Germany, and we're really trying to bring together and create a human atlas in health and disease. Thank you.
All right, great. Yeah. So I just have a few slides, but again, we'll give everyone a chance to introduce themselves as well. But to begin with, like I said, our challenge is heart and vascular. So 2 sort of separate organ systems that we're going to analyze together. And we, so fully expect that we're going to have different priorities with each of these. But for the, what is great about heart and vascular is that, unlike maybe other diseases, there's this like constant jockeying for who has the best disease. We won't get into that at the session, but for both heart and vascular, there are distinct diseases that seem to be caused by cell types and cell states. And for the heart, that's, examples of that are cardiomyopathy, ventricular tachycardia, and congenital heart disease, which is a patterning defect. And so you can imagine that single cell analysis will have direct relevance to the pathology of these diseases. And then, recently there's, been a lot of work on sort of the cellular architecture of congenital heart disease, for example. This recent Nature paper from a group in Australia showed that the stages of trabeculation, these kind of ridges in the heart muscle, the actual process is actually ingrained genetically and developmentally in heart cardiac myocytes. So sort of an interesting application of cellular analysis, and that's without single cell techniques. And so using that approach and moving to human tissue will be, I think, very, very valuable. And then for vascular, again, there's a lot of diseases that are just cellular in nature, the canonical example is atherosclerosis, so thought to be the first cellular disease. Rudolf Virchow, right, so the father of the cellular basis of the disease, used atherosclerosis as his first model of a cellular disease. Arterial dissection, hypertension, endothelial dysfunction. And just sort of the example of this is atherosclerosis, we sort of draw these cartoons of a blood vessel wall. And when you get cholesterol buildup in the coronary artery wall, we think there's like 4 major cell types: endothelial cells, vascular muscle cells, foam cells, maybe some immune cells in there. But with single cell analysis, I think this could be further ingrained. We could really understand a process like this. And so the state of the field, where are we? I mean, there's a lot of people using single cell methods. I think like with 10x and other sort of droplet-based methods, this field has been democratized, right? We all can do this pretty quickly now in our labs. But there's been no comprehensive map, I think, at this point. So I'm just going to highlight a few studies that have been done. I mean, recently in Circulation this year, a group in the Netherlands published this paper where they used single-cell sequencing in mouse hearts that were healthy and diseased here meant ischemic, so they caused ischemia, and they used very small numbers of mice. They compared the control to the ischemic, and they identified this sort of new population of cells that expressed CKAP4. That was a fibroblast population, and they sort of implicate that as a marker of this fibroblast population that occurs in response to ischemia. So it could be a very interesting finding. Is it relevant in humans? Is that what's causing disease? They have some nice data where they overexpress this marker, and it does result in increased kind of disease. They can induce expression of the marker. So interesting. And then as far as vascular, I think we all sort of are pointing towards the recent Tabula Muris paper, right? 20 tissues, one of which was aorta. And I will say that, so the number of aortic cells was on the lower end, but, very comprehensive analysis. And one thing that I thought was very cool in this paper is they looked at the transcription factor signatures. So from all their sequencing, they had 1,000 transcription factors in their sort of, 2,000 genes, or more than 2,000 genes in this case. They had a total of 2,000 transcription factors that were expressed. And on the right here, you'll see that, for the endothelial cells, they could sort of assign endothelial cells to their organ of residence based on their transcription factor signatures. So this idea that endothelial cells carry a marker from the organ that they're in. And so in both cases, these were animal models used very well, right? And but this is sort of the state of the field. Is the best maps we have for the heart and for blood vessels has been in mouse models, right? Human atlas will require more work given our access to tissues. And so I think Michela has some slides she wanted to show as well before we get started with the conversation. But we'll go from there.
I think we can use this slide just to start the discussion. Doesn't want to be a serious presentation. I think, we've seen it in several other breakout sessions. The first aim is to create a reference atlas of the heart. So definitely we need to do this. The first question is, as in many other organs, is what is a healthy heart? How can we access a healthy heart? So I think it's a discussion that it might be good to start without me imposing or starting with any, telling us what we think. It'd be nice to know what everyone is thinking about this. And then we will build in parallel developmental atlas and environmental atlas and an adult atlas. I mean, the correlation with disease and the importance of doing this is very obvious. The developmental atlas in cardiac disease has an immediate benefit and impact on congenital malformations. And the benefits of building an healthy reference atlas is fundamental to address questions in ischemic heart disease, which is the first cause of death worldwide, in cardiomyopathies, and so on. So I don't know if we want to start to discuss about what is an healthy heart.
Yeah, so first, before we get into discussion, I kind of wanted to, so, we introduced ourselves. Some of us are interested in heart, vascular. I wanted anyone in the audience, you don't have to, but like if you're working on single cell analysis of heart tissue, vascular tissue, introduce yourself, maybe say what you're working on, so we have a sense of the audience.
So my name is Norbert Hubner, and I have been leading over the past 12 months a pilot project within the CGI on single-cell sequencing in the heart. Okay. And I would like to disagree a little bit on your differentiation between vasculature and cardiomyocytes. And I think it's very good to do this, but on the other side, there's a lot of interplay. Sure. I think actually this is super interesting to address also the interplay between the vasculature and the myocardium or cardiac tissue, so to speak, because hypertension certainly also has an effect on the structural morphology of the heart. Sure. Factors like TGF-beta are actually coming from the vasculature and induce a number of changes in the heart. So I think we should capture this as well and not keep it completely as separate entities. Right, right. Yeah, that's a good point. So, one thing with vasculature is that it probably affects every organ system, including the heart. And so even for the lung, for the gut, for the immune system, the interplay with vasculature will be important. And so for the heart, it's no different. It's a good point.
Hey, thank you. I'm Loyal Goff from Johns Hopkins, and we have already generated a few single-cell data sets in CNS, retinal, and non-CNS vasculature under varying conditions of either ischemic treatment or hypoxia, but also in mouse models where we've done a number of different genetic mutations that are associated with retinopathies or vasculopathies as well. So disease is important, but also the idea that one of the interesting things here, I think, is that we can actually have models that will allow us to look at development of the vasculature in an adult system and in a model system, and those are like hypoxic conditions, for example, where we can induce angiogenesis or vasculogenesis. Interesting. Are you doing any human work also, or? Proposed, not there yet.
Okay. All right. Anyone else? No one else? Okay, perfect. So, Rick, do you want? Oh, go ahead.
So here in the YouTube, it says that Gregor Andelfin from Montreal has been working in mouse and human heart tissue, mostly development, and would like to contribute. Okay, perfect. So that's from the web server. Okay, cool. All right. So was there someone else? Yeah.
She's not here, but I'm collaborating with Bernice Murrow at Einstein, and she's working on the, she's done single cell studies on the outflow tract and developmental heart in mice. Okay.
So, sorry, the CGI project is human tissue, right? Human. Okay.
A number of people involved, like John and Cricket Seidman. Okay. And Stuart Cook, and also more on the methodological side, Nikolaus Rajewski, and Jan-Philipp Juncker.
Okay.
And then we teamed up during the course of the CGI pilot projects with Sarah Teichmann and Michela Noseda to establish procedures for tissue procurement that are working actually quite well in Cambridge. And we have addressed a number of fundamental challenges. The other Cambridge, yes, across the ocean.
Sure.
And we addressed a number of fundamental challenges because of the size differences of cellular size differences, sure. Large cardiomyocytes versus rather small cells, and multinucleation issues and things like that.
Okay, well, so you're actually, it seems like the only person who's done human cardiac tissue at this point. So, I mean, I think as we discuss more, it'll be really interesting to hear where you are, how many samples, what, those kinds of things. I think there was one more person who, nope, definitely more people. Okay, go ahead.
Hello. Hi, I'm Xuegong Zhang from Tsinghua University in Beijing. So myself is a bioinformatician and supported by a CGI pilot project on the computational side. But I do have collaborations with local hospitals in Beijing working on human hearts. We are developing protocols trying to get the single cell analysis of the explanted heart tissues.
Okay. We would, yeah. No, we'll talk about that, but yeah, right now anyone who's working on something.
We have someone up here, too.
Okay, go ahead.
Hello, Mats Nilsson, Stockholm University. I'm part of a collaborative project in Sweden where we're doing a human developmental cell atlas, and we're with particular emphasis on brain, lung, and heart. And we've done so far mostly work on the brain and heart. Okay.
Is that your paper in Cell Sneak Peek on the embryologic?
Right. So that's Joachim Lundberg's part of it, and I've been working with him to make also now spatial atlases of that particular heart tissue.
Okay. So you use developmental hearts at what stages for that study?
Yeah, it's in the first trimester.
First trimester, okay. Interesting.
So Shankar Srinivas from Oxford says that they have been working on embryonic heart tissue in mouse and human, and they are in Oxford.
Okay. Embryonic. Okay, so a lot of developmental biologists who are part of this, and it makes sense. With the heart, the developmental biology actually directly results in disease. Okay, so I think everyone's kind of introduced the efforts that are ongoing. Again, sort of early days, I think, for our tissue compared to lung or certainly the immune system. So, if we were at the end of the session to sort of have a first draft atlas, I think we would have to say, what parts of the heart would we study, and how would we get those tissues? So, Rick's a pathologist. He's gonna tell us if that's even possible.
Okay, so I think actually heart and vessel is a wildly heterogeneous, related in many ways, but heterogeneous territory. Certainly in the adult heart, we're talking about atria, ventricle, auricular appendages, or even different valvular conduction system, septum versus lateral wall, RV versus LV, all feeling very, very different effects. Even the vascular system, where you may think that large vessels pretty much everywhere are created equal, we know that there are a number of vasculitides, for example, that affect just the head and neck vessels and don't affect the lower extremities at all, or the viscera. And vice versa, we have a number of other arteritides that affect viscera and don't affect head and neck. So there's something different about the developmental onlog in those different sites. So I think the challenge with the human, as opposed to any mirroring models or other animal models, is also how we procure that tissue. And certainly if I'm holding a heart in the morgue or in the cutting room, something has happened to that patient that has affected the function and the structure of that heart. And we don't routinely, in fact almost never, biopsy normal human heart. Even if we biopsy the normal human heart, we're looking at a very restricted subset of cells. The way that is done is a right ventricular biopsy of the interventricular septum. You run in a catheter down the internal jugular and then across the tricuspid valve and into the heart. And it's a random sampling of about 100 microns thickness into the heart. So we're not even anywhere near a lot of the other interesting components. So I think there's going to be a challenge. I think that we should, from the very beginning, as we do this, embrace the fact that if we're using human adult or human well along, not neonatal or not fetal tissue, that there's going to be pathology there. And we should realize and recognize and embrace that and look at that very much in depth. Having looked extensively at the GTEx tissue samples for myocardium, for example, I know that even though they are theoretically from relatively normal, healthy people who've had other traumatic accidents, there are apples and oranges in terms of myocyte hypertrophy, interstitial fibrosis, and working with groups over here at the Broad and the Whitehead, the pathology of the samples that they're looking at a molecular level is really very germane. So I think that we also need to have pathologists, and not because it's going to be job security for me, but pathologists very, very intensely involved in both the harvest and the analysis of what those tissues look like before we just pull out single cells and try to do something else with them. So a bunch of caveats. I'm very excited about the possibilities and what's likely to happen down the road, but I think we have to go in realizing that this is a huge and heterogeneous territory that's always going to have some disease in it. The final point is that if we're going to use cadaveric tissues, there's going to be a lot of variability in terms of when those are harvested. So I have been on the autopsy service on a regular basis. I know that patients typically die in the middle of the night, and we don't even autopsy them until 8 to 24 hours later. 8 hours is remarkably quick. So that's also going to affect the quality, viability of individual cells that we want to look at.
Yes, maybe that would be a point of discussion. So
I can start to say what we have been doing. Sorry. No, you go ahead. Yeah. So healthy tissues. First question is brain death, circulatory death. We try to prioritize brain death. Of course, even in a brain death patient is dead, is the heart healthy, but that's the closest we can get. For disease patients, it will depend on what type of disease and which technique you're going to use, but we're using frozen tissue banks for single-cell nuclei and leftover atrial tissue is something that is routinely available, but of course it's only atrial tissue. And in our case, our surgeon can take biopsies from the left ventricle in multiple positions. These are very small, and, but that's for sure a very useful resource. For the fetal tissue, we have a centralized system in the UK. It's called HDBR, the Human Developmental Bioresource System, where they collect embryos and fetuses, and then we can get the heart and other organs. For the fetus, of course, as I'm pointing out, there's some phases of development are going to be more easily available. That depends on the ethics and the laws of the country you're working in. And on this point, I like to also stress something that we don't routinely discuss, is like how do we define an healthy fetus and what metadata we should acquire for this. I'd like to know what you guys are thinking. Yeah.
So backtracking to what Michela was saying about the adult source, I don't know if you remember in the presentation that I gave at the very beginning, there was like a little flow chart on the Cambridge Biorepository for Translational Medicine, which is run by Korosh, who's a transplant surgeon who's actually at the meeting, but I'm not sure if he's in this session. And what he set up is a kind of team or a system. It's also supported by Wellcome, the Trans-Zuckerberg Initiative for tissue acquisition from deceased transplant donors. And there, obviously, as Michela said, there are different types. I'm not an expert on this, but the brain death and cardiac death, essentially. And according to Korosh, like some of the hearts are still beating, basically, where he's taking the biopsies. So the ethics is for 5 centimeter large biopsies. So that
seems pretty fresh tissue.
Yeah,
then.
and they are not suitable for transplantation for whatever reason. So
it
there's
raises
an issue
If they're
there.
not suitable
Clearly,
for transplantation, they're not normal.
Yeah.
Even brain death with a normal heart has microvascular spasm and catecholaminergic in microinfarcts. So there's, in fact, ischemic myocardium scattered throughout as well. So not perfect normal.
And how have you guys, have you taken samples for pathology as well to like assess, or no, it's just sort of it's frozen tissue for RNA right away?
No, we bank for everything.
For everything.
For everything. We'll do fresh cells, we'll do nuclei, we'll bank the frozen tissue, fixed tissue, so we have an array. I mean, the advantage is if you have a resource like the one that Korosh is running, you got big pieces of tissue, so you're allowed to do this. If we're talking about a small biopsy, you won't be able to do all of these things.
Right. And on the samples that are larger, are you looking at the histology?
Yes, we fix to then look at the histology and always have those metadata. When you have tissue banks, depending on what the tissue bank has available, then you will have all the sets from the same patient. But the idea for the samples that we have from the Cambridge Biorepository and also from another source, these are donor hearts that do not find a match, we'll collect as much as we can. And trying to record the anatomical regions. And our priority at the moment is to focus on 6 regions.
Okay. Yeah, we'll probably talk about that next. Yeah,
yes.
Maybe like which regions. If you can't take the whole heart and grind it up. So
no.
Why don't you go ahead?
Hi, I had a question about metadata. And so is it possible to collect information like cholesterol levels or other metabolites from the blood that can help you maybe stratify patients? Is that useful?
That would be really good. I think it would be a really massive task, especially for the healthy patients, because you have to go and collect. It depends what mode of death, etc. We have collected a number, about 20 frozen samples from NDRI. So when I was working here at Baylor. And for those patients, we have some basal metadata from the patients. Stuart Cook, for example, has a big resource of disease samples where he collects a lot of metadata. So I think one would have to really design studies and very carefully plan and working very closely with clinicians to try and collect all of this information that I think will be fantastic.
And I would like to sprinkle in a little bit realism to the whole discussion. I think before we start thinking about cofactors and whether we can stratify according to risk factors, I think we have to address a number of very basic issues. I think it would be fantastic if we can agree already on, a set of regions that we collectively want to do, and then things come in like what you mentioned about microspasms upon catecholamine, effects on the heart, and these are overriding effects, I think, compared to risk factors that also contribute. I'm not saying it's not important, but I think we need to get a baseline here first, and of course we should also not be hindered by the fact that nothing is optimal, and we should do the best we can, and nobody is voluntarily donating his healthy or her healthy heart. So that is a limitation we have perhaps compared to other organs. And I think in terms of risk factors, I think we would need to do large numbers to stratify effects here. There are perhaps more important issues like sex and ethnicity that are stronger effects rather than in the first instance risk factors, just as a point for discussion.
I would definitely agree with that as a first pass. Age, and gender are going to be your major things that you want to look at. It's always nice to have additional data, and it's beyond cholesterol, it's inflammatory markers, it's a variety of other things. And I think it's also thinking about whether you're talking about cardiac disease or you're talking about vascular disease. And again, we're in a combined group here. But I think your point too about just embracing the fact that these are not going to be normal. In fact, expect that they won't be, and use that in terms of the analysis. If we know what the pathology is, we can actually get a lot more data out of that and make it more useful overall.
Yeah.
And so I think that, again, having a pathologist involved in sampling to an analysis of what those tissues look like before we even get into the molecular analyses, I think is key.
Yeah, I thought that was a nice point made by the lung group yesterday. So, with the lungs, right, if you just kind of ride behind a bus, right, your lungs will be altered. So if that will get taken up when they do their single cell analysis. So they actually just embraced it. They said that they would have to even start with disease states because that could help inform what they see in the normal state. And I think that'll be the same with the heart, right? So if everyone has some degree of atherosclerosis, if we take people with like actually clinically identified atherosclerosis and identify the cellular architecture of that, then we'll be better able to identify the atherosclerosis that's sort of underlying in people who weren't clinically deemed to have atherosclerosis. And for the heart, maybe the equivalent of that is cardiomyopathy, right? So we all have some fibrosis of our heart after probably age 20 at some point, or in these brain death cases, they probably have more fibrosis from ischemic death. So if we take people who actually have clear ischemic cardiomyopathy or things like that, and we then use the cells identified in those hearts, we can better understand the so-called normals. I don't know if in CGI how many hearts? All healthy, some diseased. What are you guys starting with?
So what we have done is extensive work to test it out in animal models, in mice and in rats. And then we have recently switched over to human tissues from the Cambridge Biorepository to test if dissociation protocols and single nuclei sequencing work on these human samples as well as they do in rodents. And we have confirmed this, but we have not started to produce data from in a sense of from different regions because we have piloting what is technically possible, and that we have confirmed. Okay. So interesting. So and then one last thing before we get to the sites that we'd sample. This comes up often at the other sessions. Is like, how many tissues do we need to be confident this was a good map? We can all vote, I guess, but is it 300? Is it 100? Is it 50?
Yeah, I don't think it's a good idea if you start voting for this. I would really actually like to address the computational part, which you didn't talk about at all, and I think Barbara is actually working on experimental design along those things, right? So what I was wondering is how you plan to actually, let's say, make use of existing pilot studies, such as Norbert has, or also animal studies, where you actually get some idea about the variants, to then really inform the next steps, and then to not have this sort of ad hoc discussion, should we have 10, 50, 100 or something like that, different hearts, and how many different areas do we have to sample, but to get sort of closer there. What are your takes on that, and how do you sort of plan or suggest to approach that?
I could ask you too, because you've done a lot of this work. But no, it's a really good question. I think along with everything that we're talking about, starting from essentially disease state hearts, we have to think about batch effects and just sort of looking at the metadata to figure out exactly what kind of heterogeneity we have. There are the inklings of methods to start to be able to correct for batch effects, but you know, if you have complete confounders in the data, which I assume in heart, given the number of samples that we have and the complexity of disease states that you guys are talking about, we're probably going to have a lot of complete confounders. And that's where we need to be a little bit more worried about what conclusions we can draw based on the disease data we have. Yeah.
Is your observation as well that if you go to single cell level, that heterogeneity actually decreases across populations? Because if that is the case, and I think we do see it in some other studies, not the heart, right? But if that's the case, then you might not actually need to have 300 or you know, these super high numbers. Do you have these observations?
I think at the single cell level versus at the batch RNA level, bulk RNA level, sorry, it becomes much more heterogeneous, more complicated, because we have to deal with cell types, cell states change pretty dramatically, expression levels, ischemic time affects transcription. There's going to be a lot of heterogeneity going on across samples and even within a sample that I think bulk can often sweep under the rug. There's also actually, and one thing that I realize we don't have on the list of things to talk about that I'm really interested in hearing from people in the audience who've worked on it is the technologies. Are there specific types of technologies and protocols that are essential in the type of tissues, the heart tissues we're talking about, and how can we work with technologists, we being the computational people, work with technologists to really get the most out of those data.
Can I just challenge that? Because I was hoping actually that if we took, let's say, your and my heart cells and threw them together, and maybe also some very sort of severe disease case, although I heard we all have some heart problems already. It's a good take home for me. But don't you think that our cells, individual cells, could be very similar, but just the composition sort of, I don't know, maybe vasculature, you know, like we have more of those different cell numbers. That's actually changing. If that's the case, then, you know, the batch effects, you could do alignment, you could transport labels from another heart. You understand what I mean? So it's just more population numbers than actual genetic
absolutely.
states.
So there might not be a change in, there might not be a brand new cell type, but you just get more of a ventricular myocyte or something like that. I think that's what the animal models could be informative, right? Like you said. So you know, I'd say with atherosclerosis, that definitely could be the case, that you don't get like a new kind of macrophage, you just get more macrophages that come in. I'd say with cardiomyopathy, we do think it's actually a transition to a new cell state, like there's a fibrotic cell. So again, like for each disease, it'll be different, but it's a good point. So your point is that if you're comparing just cell numbers instead of cell types, you would actually, the batch effects wouldn't be as relevant. Like
that would be my hope, that this would be an answer to not actually having so many people, but just more cells. Because you know, that's always the trade-off, right, if you have a fixed budget.
Right.
I want to make a comment about using animal models. To make the animal models useful, then they have to somewhat represent the population. That means much more genetic variability in their background. We can't just look at the same cell lines. So we need to start to look at many cell lines. We need to look at young, old. So I think it's something to keep in mind, and how much investment should we put on that, and how many numbers with the mice, for example. Would that be something that we do in parallel, and so that we can have an estimate for the long term?
I find the aging effects in single cell in general super interesting, because I think some labs, I think John's included, have shown that, you know, the variance and these type of things go up, so very basic effects, but I don't know if there's that the variance of gene expression in general goes up over age, right? John has his one paper, right? And there's, I think, some follow-ups. I guess the heart, there's some sort of more large-scale effect as well, but sure.
Yeah, there's a few more questions in the audience.
How are we going to address early cell-fate decisions in humans? I mean, we can do this in mouse. Are we going to go for organoids and iPSCs, or can we get material that early, or? You know, that's because a lot of these early cell-fate decisions will determine morphology, right? So.
Right. Yeah. So I mean, this is maybe a developmental question.
In my experience, because I've just been exploring these options, at least in the UK, For us, getting anything before 6 weeks post-conceptional week, it's very hard. Is it doable? It's not doable in a matter of months, but maybe our colleagues in Sweden that have done a lot on the first trimester, they can tell us how they got access and what age exactly you were able to profile.
Microphone.
Yeah.
Yeah, so we get specimens from Karolinska Hospital. There is a centralized facility for collect a biobank for collecting these kind of samples, and I think it's the normal something from week 6 to 12 or so.
Okay.
Oh wow.
Yep.
Okay.
So the problem is just before the 6 weeks.
Yeah.
6 to 12 weeks.
6 to 12 weeks. Yeah.
Right.
So those are the tissues more easily accessible, definitely.
Okay. So yeah. So.
Shall we? Sorry.
Go on to
one thing we haven't yet touched about is like I mean, we just mentioned, but it'd be good to discuss a bit more about the technicalities and difficulties to sample the entire spectrum, cellular spectrum of the heart, because the cardiomyocytes are really big. I mean, there I put an approximate relationship between a lymphocyte and a cardiomyocyte. So a lot of the platforms are developed keeping in mind the hematopoietic system. So the cells there are very different from the cells that we are dealing with. Plus, they can be multinucleated. So how do we get around that problem? Well, one easy shortcut is just to do single nuclei sequencing. But that is a shortcut, and it gives us a series of answers, but not all the answers. So Isolate is a platform that has been proposed to work well with cardiomyocytes. I think Norbert has experience with the Isolate. I don't know if you would like to comment about the technical difficulties with the cardiomyocytes and your experience.
Yeah, so it's difficult to address this with conventional drop-seq methods, and so we are trying to address this in different ways. For example, with Oni Basu at University of Chicago. Where is she? Is she here? Maybe you can talk about this yourself. So I cover first ICEL8, and then Oni can comment. And then, of course, well-based methods that are a bit independent of size, cell size, are probably the way to go here.
And you're still using individual cells, not nuclei?
Individual cells, then.
Okay.
Yeah.
Is there a reason you chose not to go like a
we do nuclei, yeah. But we, I think, need to do this in parallel because the cardiomyocytes, to capture them, again, with conventional drop-seq methods is difficult. So the combination of live cells plus nuclei, I guess, will give an accurate picture here.
Okay. Actually, I would also...
So just maybe Oni can briefly comment.
Can you hear me? Yes. We have started a collaboration with Norbert a few months back, about 6 months back, where we started getting adult cardiomyocytes fixed in methanol, and we have been able to successfully load them in drops, custom drop-seq devices, and also we were able to lyse them. The other thing that we should note about these adult cardiomyocytes is there is a lot of proteins, extracellular matrices that go along with it. So they are very structured cells, and lysis was a little bit of an hassle, but we were able to do it. And I should also say that we have some adult heart data also on human and chimp as a comparison, but they are all single nuclei data.
A couple points. Not only can the cardiomyocytes be multinucleated, they are, about 10%. We'll have at least 2, and occasionally more. 2, I think it's also very short-sighted to just focus on the cardiomyocytes. Although they constitute the huge bulk of the heart, of the myocardium, in fact, they're only 25% of the total cells. And the interaction between cardiac fibroblasts, cardiac endothelial cells, and cardiac myocytes is actually very germane and should not be excluded or ignored. So just looking at the cardiomyocytes is too short-sighted. Finally, they're inflammatory cells. They're innate immune cells that are probably very important in terms of normal structure and function. And so recent work from the people at Mass General looking at the AV node have found that, in fact, macrophages express connexins and are critical for normal conduction. So we need to look at everything.
Yeah. Go ahead.
So I just wanted to go back to the batch effect question and whether, I guess I'm not quite sure what we're talking about there, but is the vision that there will be multiple locations where heart's tissues are collected and multiple places where the dissociation's done and the single cell protocols are run? I mean, how many different places across the world are we talking about there? Because, I mean, that might feed into the question of how many hearts or whatever. I mean, it seems like at least it's important to say how are those going to be spread out across different centers, et cetera.
Right, yeah. So I think, I mean, it's going to be done widely. I think there's going to be consortia. There's some already. Certainly there's efforts here at the Broad to do human heart sequencing. And you know, on kind of a first guess, I guess there'd probably be 15 centers that probably work on this just right off the bat, maybe even more. And will there be an opportunity to compare data or aggregate data, or will the batch effects be so insurmountable? And batch effects are also going to be sampling, right? So the heart is not a repeated organ, right? So like the liver or the kidney, where there's different kind of parts that repeat over and over. So you can maybe, you know, even if you sample from different areas, it might be the exact same. The heart, you know, every myocyte could conceivably be different. And so my sense, and people can kind of chime in here, is I think the batch effects will be insurmountable to just aggregate all the data together. I think that will be too challenging. I do think this will be separate efforts, but will be complementary, and hopefully will provide some reproducibility for major disease-relevant markers or major, you know, new populations. But that's just my guess.
I mean, that's a little scary, right? That we can't aggregate across the 15 centers. I think, I guess returning a little bit to Fabian's question too, you know, we sort of have this idea that we have some number of cells, and that makes up for the fact that we only have, you know, 200 parts, samples from a particular region of the heart. And I don't think that there's a really clear mapping yet, at least in my head, between the increase in the number of sort of these independent samples, such as the cells, and the power that you get from having so many of them, especially when you can aggregate across different centers that are collecting the data. So I don't exactly know what the power trade-offs are, but I also think it's worthwhile to actually start developing approaches to make those connections. Yeah.
So Rick, can I ask a question to Rick? One of our always discussion now we have with Sarah and Norbert, we try to focus on 6 regions. So we focus on the free wall of the left ventricles, the apex, the interventricular septum, the free wall of the right ventricle, and the 2 atrias. So it's, you might say it's quite vague, but what can you tell us? How can we standardize at best the way these pieces are sampled, and how can we then compare? So can we come up with some anatomical points and coordinates to tell us, well, we get that piece as close as possible? And is it something that we feel it might be of any help?
Sure. I mean, I think that once it's decided what exactly you want to look at, then you can somewhat standardize that. And I think a mid-transverse section through the myocardium, so kind of halfway down from the base to the apex, and a mid-transverse section, you could look at lateral free wall, septum, and RV in that plane and do that reasonably routinely standardized. I think that you can definitely look at AV node. And I think that you can look at AV nodal tissue. I think looking at all 4 valves is not as, I think looking at one valve, and you could routinely look at the aortic valve. And maybe mitral, I take that back, because there's myxomatous degeneration in the mitral, there's calcific degeneration in the aortic valve, and they tend to be different, so it would be interesting to look at both. And in terms of the atrium, pick your poison. Actually, so it's been demonstrated that there are a greater number of stem cells, myocardial stem cells, in the atria, more on the right than the left side. So I think that that would be something else that we would want to go after. But I think there are probably 6 sites overall that you could do. I think that you can standardize it to some extent, and that you could sample routine. But that's holding the heart. If you are doing it by biopsy, I think that now it's much more of a crapshoot. So I think that you are going to be very constrained in the number of cells, the type of tissue you can get, and the depth that you can go.
So actually, as someone who will think about doing downstream analyses of these data, one question that I had for people like you is whether we can develop some kind of coordinate framework, map both living and dead deceased hearts to that coordinate framework, and then reliably be able to place each of the samples on that coordinate system.
So as opposed to an animal model where they're going to be relatively homogeneous, every heart is unique in terms of that. I mean, you can try to get coordinate and map it appropriately. I think that you're going to be off to a variable extent. I think that you're going to have to live with that kind of variability. A little bit of dilation, a little bit of hypertrophy, a little bit of fibrosis, relative predominance of a right or left ventricle. I think it's going to be quite difficult. Even looking at trabeculae versus muscle that is not trabeculated, I think is also going to be different.
So what types of questions about transcription are interesting in the context of the spatial structure, the morphology of a heart? Are there interesting questions about gradients of gene expression or cell type or structure related to the framework?
Well, I think developmentally, I would say that that's absolutely key in terms of gradients. I think that that's going to be a very critical sort of question. In terms of the adult heart, again, it's related to the pathologies and interstitial fibrosis, endothelial cell dysfunction, cardiac hypertrophy that is not uniform. Even in a hypertensive heart, the left ventricle is not uniformly affected by that. So it's somewhat unique depending on the geometry of the heart. Again, as the heart contracts, it doesn't actually, it's not just a tube that does this, it's a squeezing, very complex 3-dimensional motion that affects individual cell activity. So even that, the kind of where you are in terms of apex to base, in terms of that squeezing motion of the heart, there are probably going to be gradients of difference in terms of expression of certain myocardial sarcomeric structures.
I would add to the interesting pathological changes you just alluded to, also in the healthy quote unquote heart. I think this is already quite interesting for cellular circuitries. If we can do spatial resolution within the heart, which cell is secreting what factors, and that affects the program of other cells, fibroblasts versus cardiomyocytes, for example. And I think we can address these questions with the appropriate analyses in the heart, even without comparing to disease.
So another question this brings up in my mind is we have actually a lot of imaging data of the heart, so live beating hearts, MRI type imaging techniques. What role do those play in this analysis? Can we bring them in at all, or are they just sort of something we can't use?
Well, I think actually having functional data on anything that we're going to eventually analyze is key. So I'm looking at contractility and whether there's non-homogeneity in terms of the wall motion is critical. We won't have that information, however, on the vast majority of what we're looking at. So the exceptional case, we should be all over it and look at every single cell in that heart, but it's most of the time we won't have that data.
So you're saying essentially use it for phenotyping, right? So understand sort of at a gross level what's happening with that heart.
Right, no, to see what the ejection fraction is, to see if there's non-homogeneity again in terms of the wall motion, looking at valvular motion. So I think there are a number of things that we would love to have on every single specimen. You're not going to have that in the vast majority of cases. Even conduction system mapping, it's unique for every individual patient, and that will probably also influence local gradients of transcription factors.
I mean, I will say that it could be possible. I mean, we're talking about a limited number of hearts, and let's say we limit it to all sort of brain death cases where it was rejected for organ transplant. So all those patients would at least have a cardiac echocardiogram, probably not a cardiac MRI. And an echocardiogram, so what information? Definitely EF, and definitely sort of questions about is there any sort of wall that was ischemic or akinetic or something like that. Certainly the reason it may have been rejected for transplant, it could be from the echocardiogram that was done. So that information would be there, but I don't think it would provide like a common coordinate system or something that you could map the biopsy back onto, unfortunately. Right, of course. There's non-cardiac reasons why they're rejected.
Yeah, I mean, exactly. I mean, sometimes they're complete. So again, I'm not an expert, but my impression kind of from working with his transplant kind of system in Cambridge is that sometimes there are regions that are completely unrelated to the heart, that the people who are responsible for that just don't want to take it, like an infection in a completely different part of the body or something. Right.
So is it unrealistic to try, given the complexity of the structure of the heart, is it unrealistic to try and build a spatial model in a non-human model system and then try and map that? I mean, can we spend concerted effort trying to get that geometry, that coordinate system put together there, find marker genes for each attribute that we're interested? Is that where we should be spending effort at this point?
I would say yes. I think that you're going to be able to have much more control, better quality material, and be able to look at the very subtle things that we've been talking about in animal models. And then you can try to see if there's correlations if you sample wherever you're going to sample in the human to see whether they're kind of you're comparable.
And is there a preferable model system for the structure, physiology, et cetera, of the heart that should be the focus for the human cell atlas?
The animal model that's most comparable is pig, is porcine hearts. Those are the most comparable, but actually that ups the ante. Virtually every single cardiac myocyte in them is multinucleated. But physiologically, they're the most human-like.
I know there was someone who said that they were working with, I guess, monkey or primate hearts. I guess maybe not here anymore. But I wonder if those hearts are more, I guess, consistently shaped and sized.
They are, although there are some interesting problems associated with primate aging. That there's an interstitial. I mean, it's great and potentially germane to human disease, but they get a progressive fibrosis, interstitial fibrosis of the heart as a function of age. Okay. So there's pathology that just happens intrinsically. Would be interesting to understand that. But so primate hearts, I think, have their own issues, and even getting them is also a bit problematic. It may be easier to get a human heart than some ape hearts.
Got it.
As a word of encouragement from a computational point of view, I don't think it's unheard of. I mean, we can morph gels from one to another. We can morph images from one another. We could morph common coordinate systems. So if you had relative distances in a single heart, you could then morph it into another heart and then have the positions of the myocytes versus another in some sort of relative framework. So I wouldn't discourage it. I would say that it's going to be just another layer of challenge, right? So.
Mm-hmm. I agree.
I think we should think and try that
Sure.
in some way, because we're always going to be, we are a continuum of differences. No one is like the other, but we could try and rebuild a virtual heart.
That's some the case.
Yeah.
I mean, there's another organs that need this.
Yeah. So we didn't come out with a number like it happened for the lungs yesterday.
Right,
they said
yeah.
100.
Well, they were pushed. They said, well, we don't know, and then someone else said 100, and they're like, great, 100.
So do we feel that the 20 hearts or the 20 donors that was discussed originally in the HCA is going to be sufficient or not? Do we need to aim much further than that?
And how do we actually sample those hearts too? What
Exactly.
regions, how densely? Yeah.
Mm-hmm.
Yeah.
Mm-hmm. Yeah, so I guess like the sense I'm getting from the conversation so far is that I think we're very interested in sort of a lot of breadth to one sample. So multiple samples, multiple locations from one heart, as opposed to maybe one sample from hundreds of hearts. And then it sounds like we might have to be iterative. So we don't even know what the variability will be from sample to sample. And so that maybe is the more sort of, the less arbitrary answer than like just coming up with a number, is that, you know, as we start to do this analysis, we'll get a sense of what the inter-individual variability is based on age, it sounds like gender, ethnicity, and biopsy procedures, organ obtainment. And we're guessing there's going to be a lot of heterogeneity, but we probably with 20 will have a sense of that heterogeneity.
So maybe instead of keeping going for numbers, we could go for time. Like, let's see how much we can do in the next 2 years, and then from there on, we can, we'll keep reassessing along the way, but maybe I think that's a better point. To reassess and really evaluate what people have been doing across the world,
Sure.
because everyone is going to be doing it. And it's important that everyone does it to sample environmental, genetic, and all the sorts of differences that we'll encounter.
Right. At some point, the Human Cell Atlas, there should be sort of a procedure for aggregating the data, and we'll see. You know, maybe it is easier than we think.
So I'm a bit confused because you mentioned about 15 different locations. I mean, I think geographic locations or labs doing these experiments, and now you're mentioning 20 hearts. So I'm assuming now you're talking about doing all 20 in one place. But is that right? Or
right.
You're going to do 20 hearts spread out across 15 different places? Yeah,
it's a good point. So I guess we're sort of being loose with this because we don't actually know what's going to happen. But I think that's the truth. But I think for any individual kind of effort to claim that they can create a comprehensive atlas, we would guess it would take maybe 20 hearts to start.
I mean, the 20 organs, the 20 replicates, let's call it, it was what they were originally the HCA has said. We need about 20 organs to get a rough and good idea of what's going on. So,
okay, so let's suppose we start there and say we're going to have 20 done. There's a question of who's going to do them. Or is that already settled? I mean, are they going to be done in the same place?
I think this comes back to what Raj was saying. People are doing it, so inevitably we'll go more than 20.
Absolutely.
That's why I'm saying that instead of going by numbers, which should give us time, 6 months, a year, and 2 years, and see how many we got and what informations we got for this, because it's also new that it's very hard to plan and predict.
So isn't the benefit of having meetings like this and discussions like this to try to uniformize protocols across those different locations as much as possible? So, I mean...
So if we take Raj's comments that we would like to have perhaps donor hearts that were refused for whatever reason, to get the largest number in a uniform population would be in major centers that are doing heart transplant. Yeah. So it's got to be, and I can count on 2 hands pretty much where that would be in the world. And then it's just getting those centers when they have rejected hearts to acquire as much information and sample pick 6 sites and do that in a routinized way.
Yes.
But it's not going to be done in the middle of Kansas and in the Florida Panhandle. It's going to be done in major transplant centers.
Yes.
I mean, I completely agree about standardization of protocols and coordination. So I think what Michela also said with 20 is what we think within the people we have initiated working with, that this is a realistic time, a realistic number, 20, that we can actually procure over a period of 2 to 3 years, okay, and analyze at this stage with the availability of healthy, quote unquote, I still call it healthy, but I completely acknowledge all of what you said. With what we can procure from the sources that we have established access to.
Yeah, hi, over here. This is very far out of my area, but I'm impressed that with hearts obtained in this way and consented for open, that even a relatively modest number like 20 is this enormous resource. And just as you were thinking about doing power iteratively, which is clearly correct, the idea that this resource and keeping them so that you can return again and again to either more areas or an adjacent area, because the platforms are going to change. And if you find that there's more change from, say, 3 centers that are actually making measurements than you can cope with, you can re-strategize and come back to nearby areas and hit them again with just one protocol. And as the spatial things get better, I am, perhaps it's hubris, but I'm confident that you'll be able to do things that you won't do in the first round. So is there a provision in the plan for having these hearts, even after the first sample is taken, to be a resource to return to for the entire HCA heart group?
So I would throw it back out to the audience. What can you do with fixed tissue? So if we can formalin fix anything and keep it reasonably well preserved forever, but I don't know what...
But what about cryopreserved freeze after taking the first?
Yeah, I don't actually, so cryopreservation in large tissue like that, even in valves, is extremely problematic. The best cryopreservation techniques do not preserve viability. They do not. So cryopreserved, and cryopreserved valves are thin structures. Mm-hmm. You would think you'd be able to get that very well controlled. And basically...
So people have actually tried that for RNA and it's come out badly? Because I've seen just the tip of an iceberg of a little bit of data, and I'm not sure it's correct. But so there are things that can be done from fixed, and that would probably still be worthwhile, particularly because all the spatial things work fixed. And so things you learn from wet can be projected onto spatials done later. So that alone would justify it. But the question of whether to try to keep a little bit, either snap frozen works pretty well for many of these things. Nuke-seq out of snap frozen.
Sure.
Yeah, that is definitely doable.
But, I mean, you would only snap freeze certain areas in the heart. Yes. Again, you're not going to do the entire heart. So again, it's a sampling issue. I like the idea of being able to revisit the same specimen with improved methodology later on, but we're kind of constrained in terms of doing individual single cell, what you can get out of fixed tissue, I think.
Yeah, but the spatial stuff, you're looking at individual cell level resolution as the techniques improve, and you can probe for,
Sure.
Depends on the number of genes, but it can go very high. It will in the period of this project.
I would like to comment a little bit on the fixed tissue. We have been looking at FFPE tissues a little bit, and it seems because we do typically for high throughput single cell sequencing 3' end counting, fragmented RNA is not that much of a problem.
Okay.
So maybe I'm being overly optimistic, but it is not such a crazy idea to even think about FFPE tissues in the future.
Other people probably ran across, there was papers on doing this in cancer and formalin fixed tissues, and they could get copy number out of it. Mm-hmm. Yeah. So.
Right.
get it. The heart's I get it. The heart's the most interesting, of the organs that we've charged with talking about. But is anyone working on vascular tissue at all? Is anyone doing vascular biopsies or samples? And are there certain vascular tissues, or how are you approaching? I mean, certainly the vascular tree is even more complicated in some ways. So how would you approach sampling in that organ?
So you're talking about large vessels.
Any vessel. So yeah. So right. So how would we go about that?
So I guess we've done some vasculature in mice, and it's primarily been through sorting enrichment of vascular endothelium from reporters, which is obviously not going to be feasible in humans. But I do have a question, if we're diving into this at this point, as to whether the field thinks it's more appropriate, because you have vasculature obviously in different tissues that are going to be responsive to different types of dissociation and disaggregation. Do we make an effort to optimize the dissociation for each tissue and then get the vasculature that we can that is probably changed? Or do we make an effort to homogenize our dissociation methods to preserve what we can about the vasculature at the expense of being able to capture different things in different tissues?
Yeah, that's a great point. So I guess for large vessels, and I'm sort of doing this also, so I've done sort of mouse aortas, and I take the whole aorta without flow sorting and looked at all heterogeneity of cell types, and I sort of optimized the dissociation protocol for a large vessel, and I used like a collagenase hyaluronidase dissociation for 30 minutes, and then the reviewer said you have to use elastase, so then we tried that. I mean, so even when you so-called optimized protocol may not be optimized for every cell type, even in a large tissue. So I guess like my answer is that I don't think there's much value in standardizing these dissociation protocols, because different protocols will have different value. And so I think you can learn some things from some organ optimized dissociation protocols. So I think that's like the fun part about anyone who's interested in vascular, is that there will be vascular tissue from every other sort of single cell effort that's done, including the heart. There'll be endothelial cells from the lung, the kidney, from every tissue. And so analysis of those endothelial cells will give us some look at the small vessels, maybe even some medium-sized vessels, and then large vessels can be done sort of separately with different protocols.
There are, as you mentioned in the introduction, fundamentally different properties in different types of vasculature.
Right.
And if this is passenger information from some of the other atlases that are being generated, I mean, it's perfectly confounded. It always will be perfectly confounded, and there will probably be things that will not be comparable in those contexts. Should we have a sub-aim or a sub-goal to try and have a standardized collection method or focus on nuclei again, enrichment is not possible here.
Right.
I don't know how that problem is solved.
Right. Yeah, it's a good point. So when you say standardized protocol to get the most endothelial cells, for example, from a
not necessarily the most endothelial cells, but at least a consistently stressed population of endothelial cells from across the human cell atlas.
Sure.
Because otherwise, we will always be perfectly confounded by the protocols.
Right. If we're comparing the liver and the kidney, but they use totally different methods, then we can't ever compare those. That's a good point.
I think, sorry.
Go ahead.
I think exactly what you said, that the first step is getting the nuclei. So the protocols for nuclei isolation is quite uniform across different tissues. Then you could enrich for the endothelial cells. As with the protocol or antibodies or whatever you can that can be maintained as uniform as possible. And then you go back with spatial transcriptomics to verify what you have found. So you need, I think, a multiple approach to evaluate, identify the location, and validate what you have found.
So also just to extend on what you said about the spatial point here, should the vascular atlas be the encompassing coordinate system? I mean, is that the place where we should build the full coordinate system for the entire human body atlas?
I personally think there should be at least 2-3 techniques that are uniformly used. I'm talking about nuclei versus spatial transcriptomics that are the same across, because technically they are the same. The processing of the tissue is the same. So that would be a really good way to uniform and be able to compare more easily across organs.
Yeah, I agree. I think that's exciting, is that the common coordinate framework could be endothelial cells. I don't know if any other cell in the vasculature, but endothelial cells are known to sort of have a resident signature. I don't know if it's going to be that exact. Like, for example, if I gave you a biopsy of tissue and you analyze the endothelial cells in that biopsy, would you be able to tell exactly where it came from, or would you be able to say it's from the heart and that's what you can tell? I don't know. I mean, that would be something that we could test. Like, could you tell me that those endothelial cells were from the LV? Maybe LV endothelial cells are different than RV endothelial cells. The apex of the heart versus the septum of the heart, probably, I don't know if you'd have that much that granularity just from endothelial cells.
Well, the study in the brain have shown very
sure,
nicely. There were
true.
gradients of endothelial cells, changes in the smooth muscle cells in the pericytes. So the study in the brain really gave us an idea that there might be even more heterogeneities that we were thinking before, because a certain level of heterogeneity at the microvascular level is there, functionally, morphologically. I mean,
correct.
You guys know better than...
Well, and also, as you're aware, even in the same general location, flow differences in shear stress forces, turbulence, nonlaminar flow, also influences gene expression dramatically.
Right.
So you could be a millimeter away and have a very different signature, even in the same general territory.
Right. That's a good point, too.
Not necessarily a different type, but a different state, to just reinforce what you said.
But I think any sort of vascular project should sort of investigate that. Is can endothelial cells or vascular cells serve as a common coordinate framework? I think that would be an aim.
So the discussion made me wonder whether there's any, I think this is really challenging from a practical perspective, but any attempt to try and sample the same organs from, different organs from the same people, talking about the relationships between different organs. I mean, I'm assuming that you get a heart, and that's what you get, and it was rejected for transplant or something, and so you do experiments on that heart, and you don't have access to everything else. But I mean, you could imagine great value in a data set where we had organs from the same people. So again, it's really a practical question. I've got no idea what's even possible or what the easiest way in there would be, but I was wondering if it's been thought about.
Maybe Sarah, can you tell us in the case of Koroush, how often you managed to sample organs from the same patient?
Yeah, I mean, that's routinely the case from the same, I mean, again, that ethics at the moment is for everything that's accessible through the transplant incision, which is from the neck to the waist. And it's basically anything that's not transplanted, so that gives you an idea. Yeah, and we routinely have multiple tissues from multiple organs and so on. Does that answer the question?
Would you say half of the patients you will get 5 or 6 organs from the same patient, or 90%? I'm just curious of the numbers.
No, I mean from everyone you get.
Will manage.
Yeah.
And I know, Orit, there's some efforts here at the Broad to sample multiple organs from single donors, right? There's.
Thank you. So I work very closely with Christian Ardley, and we're leveraging actually the previous GTEx pipeline, which is based on NDRI, and it's a very nice pipeline for deceased donors, both for organ transplant and for rapid autopsy. And you can get several different organs from the same donor, for sure, and they have quite a lot of information and metadata that's tracked very nicely. So it's a very nice and robust pipeline that would be very useful, I think, to use. And Barbara, if you want to add more to that, that would be.
Yeah, I completely agree. I guess in my mind, one question that's coming up while we're discussing multiple organs in the same individual and also vasculature more generally is whether we're going to take vasculature samples across, let's say, one individual, a GTEx individual, for example, for the express purpose of mapping vasculature in an individual, or whether we're just going to get vasculature as an offshoot of collecting another organ. So is this going to be sort of residual data, or is it going to be collected in its own right? And from my perspective, in terms of analyzing the data, it would be, I think, very challenging if it were part of a specific organ collection, if it were sort of the residual data that we got from organ collection, because like everyone has been saying, the protocols are wildly different. The individuals are probably wildly different unless we went to GTEx, et cetera. So do you have thoughts on that?
Yeah, so it's not something I thought about until today, to tell you the truth. The human tissue that we have access to, I haven't been getting it from autopsy samples. So I'll tell you, sort of, I'm interested in the aorta. So cellular heterogeneity in the aorta is interesting, and one thing that we had thought about is when people get coronary artery bypass surgery, they have punch biopsies taken in the proximal aorta, and so those punch biopsies are discarded tissue, and so that's a source of tissue that we could use for single cell analysis. At the Brigham and Women's Hospital, there's 300 coronary artery bypass surgeries done a year, and they do 3 of these punch biopsies on average. So that was one source of tissue, but there'd be wide heterogeneity. I mean, different locations. They're sort of chosen ad hoc by the surgeon. They're not sort of sampled, based on our recommendations. Also, there's valve surgery that I've sort of talked to Rick about. Is every time a valve is replaced, there's some proximal aorta that's excised with that. And that could be a source, but that's again disease tissue. So that was sort of my, sort of sampling of vascular tissue, and so it was sort of dedicated to getting vascular tissue, but it was at like one point in the vasculature, not sort of the whole vascular tree. It is exciting. I think you could ask different questions by sampling the whole vascular tree. And so you could use residual data, and there's technical artifacts, but I agree that maybe the nuke-seq would be a way to get around the technical artifact, because that is a sort of consistent protocol to get nuclear, and vascular cells don't tend to be multinucleated or have kind of caveats from using nuclear-seq. So that could get around that. I think that would be more feasible. There are only going to be a few instances where we could actually sample kind of like from the aorta all the way down to its branches, all the way out to the capillaries from a single individual. I mean, that'd be extensive sort of surgical dissection and processing that I don't even know if sort of something's been done like that.
Not on a routine basis.
Yeah, or I mean, even bulk-seq RNA methods, right? I mean, I don't know if anyone's done an experiment like that using bulk-seq.
Not that I'm aware of.
Right, yeah. Just sort of maybe technically too difficult. All right. We've sort of glossed over some of the computational hurdles, but you're interested in batch effects in particular and methods to sort of validate that. There's a lot of computational people here. Do you want to sort of take a few minutes to kind of talk about your approaches or questions that are outstanding that interest you? Barbara?
Yeah, sure, absolutely. So, I mean, Matthew's also in the audience, and he is also he is an incredible statistician in the sense of thinking about batch effects in these particular data sets. So I hope you'll speak up at some point. But okay, so I guess broadly, if we're talking about vasculature or heart or either way, I think one thing that comes up in particular with respect to the heart, for me at least, is the idea of an atlas is not just a description of points in the tissue space, but really getting at spatial resolution. And again, when you think about the heart as an organ, it's a very dynamic organ. It obviously moves in these very interesting and person-specific ways, and there are going to be interesting stories to tell with respect to disease about dysfunction in this organ with respect to the dynamics, right? So I guess more than other organs, I'm really interested in thinking about this atlas as a spatial, as a dynamic spatial description of the organ, thinking about transcriptomics, but thinking about all the other aspects that come along with it. And in that regard, I'm struggling thinking about 20 post-mortem hearts, thinking about how we're going to do this properly. I realize it's a really hard task, and the statistical methods don't entirely get to the point. But I'm hoping that 20 is going to be the beginning, because I feel like we need to prospectively iterate on the design of the study, given heterogeneity, given the goals of the project, where I think, again, with heart in particular, we can flesh out really what we mean by atlas spatially in an exciting way. Given the specific types of samples that we get to, whether it's mostly male, mostly older, whatever the phenotypes that we get, make sure the stratification is there. So in terms of batch effects, I think along with this perspective idea, we need to take those into account very carefully, be clear about what information we're not going to get from those 20 hearts, right, because of batch effects, and then do a very careful job of adaptive design of the choices of hearts that we make moving forward to minimize that, to minimize the batch effects, to maximize statistical power in whatever objective we have for the atlas, broadly writ. Do people have thoughts about this? I don't want to, I feel like I talk too much. Yeah.
Yeah, so I mean, Barbara, and I'm not sure if Matthew said anything, but I completely agree on the need for thinking through experimental design, standardization. I think that the discussion about kind of beacons and markers spatially is super useful, actually, and that will be extremely helpful in this context. As I said yesterday in the gut, I'm actually feeling kind of more optimistic about this. I mean, anyway, I come at it from the experimental design from this angle anyway. I am feeling more optimistic about batch effect or integration methods, let's say, because I think the computational community is making huge progress on that front. On the front of in terms of what you're saying about skew of donors, just from a practical point of view, I mean, it's going to be hard not to avoid that. Speaking like from the program of that we work with, it's definitely skewed towards older. I mean, there are young transplant donors, but because people die, people have accidents and die at all ages, but it is definitely skewed towards older. And then the other thing, it's definitely skewed towards male for some reason, and I think that's to do with consenting by the families. So the families just tend to, for some reason, like it's biased towards men or the families consent, basically, as opposed to women, and it's definitely statistically significant and quite surprising.
That's true. Yeah, so GTEx is 2/3 male, 1/3 female, and my understanding is that's because men consent their wives less than husbands. Men consent their wives less. Even though the exact same number of donors were requested from both genders. So, yeah. Do you want to say something about that? No. Okay,
no,
so
but it's correct.
It's and actually also looking across GTEx too, just because that's what we have experience with. I think also the mode of death is really fascinating. The gender skew is interesting. The age distribution is fairly bimodal, actually, right? It's sort of, I'm trying to remember. It's skewed towards older, but we have some cutoff too. The oldest is
70.
70, yeah. Okay, yeah. So I think this is interesting in the context of thinking about how to incorporate, stratify, especially with only 20 hearts. Something that actually comes up too in GTEx that we've seen is that maybe this is too far in the weeds, but if you have multiple sites from a single individual, so we have on average 18 different samples from a donor in GTEx, it actually turns out that when you think about sort of cross-tissue mapping, I'm thinking about this specifically with respect to vasculature now, you have donor-specific confounders that you really need to control for as well. So it's not batched, but if this person is a smoker or whatever their lifestyle has been, you're going to see signatures of that across the multiple samples from the same individual, and taking that into account is actually a really important thing to do without removing the signal from this heterogeneity from this donor more broadly. You have to take that into account, though. I want to go back to Sarah's point about experimental design and sort of how we can start a conversation between the experimentalists and the statisticians. How is it best to talk about designing these experiments prospectively, choosing donors, matching samples, and deciding on protocols? How can we start doing that? Bad question. Yeah. So do people have thoughts on that from the collection point of view or the protocol point of view? Are there specific things we need to try out with respect to any of the protocols? Are there specific patient types that we need to get at immediately to try and understand disease and the atlas more generally?
Well, again, I think that if you're looking for the best quality material, that you're looking at a rejected donor. So I think you're looking at a particular, and you take whatever you get. Because you also want all this other additional data, you want to have the metadata, and you want to be able to look at functional data, and you want to be able to put the whole thing together. So, it would be nice to have 20-year-olds who all die on the same time, and we can look at their hearts in a standard way. I don't think that's ever going to happen.
Yeah, I'd say that, so some things that I think this session we could think of standardizing are the type of heart. So it sounds like there's a broad consensus of sort of, post-mortem, not sort of clinical biopsy samples, but post-mortem hearts, brain death as the sort of, or rejected organ transplant, brain dead and then rejected for organ transplant.
Right.
Acknowledging that there'll be some disease, but that's as good as it gets. Then we could standardize sampling sites. And then as far as tissue preparation, so right now I think the sort of standard seems to be nuke-seq for clinical samples, given that you can flash freeze, you preserve RNA, you get around the issue of different cell sizes and sort of issues with that. There probably is a role for also doing cellular analysis, it sounds like, for maybe you get different data from that, but it seems like maybe at least those things, the type of heart, the sampling sites, and the tissue preparation seem relatively standardized. I also wonder if, there will be groups that are focused on the normal heart. There are going to be groups, a paper is certainly going to be published where they compare dilated cardiomyopathy to normal, or hypertrophic cardiomyopathy to normal. And those will probably not be exactly as standardized, right? Because those will not be healthy donors, and those will be people, those will be the explanted hearts that are going, the patients who are getting transplanted in the explanted heart. There'll be much more variability in those samples, and I think we'd probably just embrace that as it'd be an interesting study. It'd give perspective on the normal hearts, but there would be inter-individual variability that would exceed what we would imagine from normal hearts.
So ideally, those would be the two extremes.
Right.
No, ideally, I mean, theoretically, they would be the two extremes.
Right.
And then we need to find a way to build what's in the middle, because it's good to know what's at the end stage when you have the transplantation, but you think you want to know what led to that if you want to stop it. Mm-hmm. So then going back to biopsies and as much as we can, it will be.
And at the same time, I think that some of the comments that were made earlier that we should really take advantage of animal models, and because we can do those in excruciating detail and get a lot of information and look for correlates in the human hearts when we're looking at, and then think about what we can do with fixed tissue that we can go revisit with additional techniques later on.
So a related question that I have is, what's the role of differentiated iPSCs here? Do we care about developing those systems and using them in the atlas?
I think it's a really interesting question. I mean, there are groups that are doing that. The Seidman group has been using iPS cells and differentiating the cardiomyocytes. It's only a part of the story. And they're not perfect correlates, even when you engineer in the same mutations. They do not behave exactly like it does in the context of a heart. So I think that you would lose information about fibroblast interactions and endothelial cell interactions, the total metabolic environment. I think there's utility in doing that. I think it's really interesting to do it. It's more manipulatable, but to make that the gold, it's not even a bronze standard.
I think it's manipulatable, sorry, but it's also, on the other side, it would be good as a drug screening and other type, potentially.
Yes.
Potentially. I mean, for us, it's the best thing we can do in vitro at the moment. It's the,
correct.
In terms of human, it's human IPS. We know they're not well differentiated, and there are a lot of problems starting with.
Right.
But it's the closest
exactly.
that we can, and then we can move on and build organoids and bring in the other types of cells. So there is some space.
If I may add to that, my name is Gert Vingstra from Nijmegen. We're working with Robert Basier also to differentiate those cells towards cardiomyocyte lineages, which in the process also generates other cardiac cell types. And they follow a sort of normal developmental path, going through mesoderm and then to cardiac progenitors. And the goal is indeed to get to disease modeling in a variety of ways. And you can manipulate the cells, obviously. And you also can generate engineered heart tissues out of that. And so one of the goals there is also to get to more mature stages of in vitro differentiation. And for that, the comparison with in vivo tissue is really important. So I think, yes, there will be some insights that come from that as well, but it is a two-way street, in effect.
So is there someone doing both at the same time, in vivo tissue and differentiation, so that they're done kind of in the same lab to make that comparison?
Yeah, I was just saying that we're aiming to do that, yeah, to do it at the same time.
How do you compare, how do you think about cross-analysis of the two?
In terms of cross-analysis, we try to generate the single-cell RNA sequencing data set. For the heart tissue, we're not there yet. We probably have to move to nuclei for that. But then we also would need to generate the nuclear data sets also for the in vitro tissues.
I would like to add that we had a CGI project for the Human Cell Atlas, the pilot round, where we did iPSC-derived cardiomyocytes, single cells, single nuclei, and then adult human heart tissue, single nuclei, just to do this kind of a comparison. And the data should be going up pretty soon. The one thing that I wanted to add was, if we are going to do heart tissue from many different sources, can we please note RIN scores if possible? It's not easy, it's not fun, but it is very, very useful in measuring that.
All right, so yeah, I mean, I think we have, what, a few more minutes left? Yeah, 15 minutes, 10 minutes. So maybe with the last 10 minutes, we'll say that, sort of summarize some of the things we've discussed. And then I did want to say that, though we've sort of focused on the challenges, I think, heart and vascular are sort of unique organs in this sort of single cell program. And I touched on this at the beginning, but there are some specific advantages to studying these organs that I think other organ systems, don't have the luxury of. I'd say that, first of all, with vascular, I think there is this prospect of it being a common coordinate framework for the whole body. We will get data that's informative from every sort of effort that comes of this. And I think that's exciting. I think that's something that we could take advantage of. Also, so with vascular, it's one of the few organs that's so plastic. So, the immune system's like this, but the vascular cells respond to local stimuli like shear stress or flow, or inflammation, or diet, even. And so that, sometimes we wonder about how do we integrate exposures when we look at the transcriptome. And a challenge and an opportunity with the vasculature is that we could do it with vascular cells. So if we got information about things that people were exposed to, whether or not it's infections or diet or risk factors for disease, that could be, we could measure that from single cell analysis of vascular cells. So I think that's exciting. And then again, just sort of touch the diseases that we're studying with heart and vascular are diseases of cell state, diseases of cell patterning. And so, congenital heart disease, I think, will be an immediate sort of application of some of the developmental work that's being done in this room. And then again, so while heart tissue is sort of tough to obtain from clinical biopsies, vascular tissue is ubiquitous, right? There's lots of vascular tissues that are biopsied or excised or come with other tissues when they're excised. So another sort of opportunity, I think, for single cell efforts, if they can be standardized, of course. So those, I think, are unique opportunities. I don't know if there's other sort of unique things that have drawn people in this room to the heart in particular or the blood vessels in particular. Was there any sort of reason that any of you latched onto these diseases or these organs? Maybe not. Or just assigned to this room or something. This is the biggest room. So yes, okay. So go ahead.
So related question. I hope someone should say something. But are there specific technicalities or challenges that the heart and vasculature pose that we haven't covered in the last 2 hours? Anyone have anything in particular in mind from their experience?
Yeah, right. In some ways, our challenges are the same as every organ, right? So standardization of protocols, batch effects, inter-individual variability. I think our unique challenges with the heart we've discussed are that it's not a patterned organ, so every cell could conceivably be different than every other cell. So even our sampling methods will have to be even maybe more exact than they are for other organs. Other sort of unique challenges that we've discussed in this session are the fact that every heart's going to have disease. We'll never get our hands on a truly healthy heart, unlike maybe other sort of other organs where it sort of comes with a tumor or something like that. And then I think those are kind of the main unique challenges to the heart. Go ahead.
Yeah, so my question is about the type of research people will be doing downstream with this. And again, since these will be a lot of diseased hearts, is there a type of metadata that might be more important for, say, the heart than any other type of organ based on the type of research people might be expected to do with it?
Right, yeah, that's a good point. So if we initially just sort of get a collection of so-called healthy hearts, the metadata that we could then kind of go back and look at would be pathology, I think, would be the best, right? So if there was concomitant pathologic investigation of that tissue with single cell, then if you find some sort of interesting signal, right? There's this cell type that's not in the other samples, then I think the first step would be to go back to the pathology and see if there was something that could be, examples of this could be fibrosis or, some hypertrophy, things like that could be found. There's preclinical imaging, so like we said, echocardiogram, but cardiac MRI, you could, again, find hypertrophy or fibrosis. It's just sort of a different kind of pathology there. And then beyond that, those are I guess.
Yeah, is whole genome sequencing something that we have to keep in mind and not to forget? So if we don't have the resources to sequence right away, we should keep some aside and make sure that if we need, we can go back and check for that as well.
You were asking about potential unique features of the heart. I'm not sure if it's unique. Well, it is in my heart unique anyway, but plasticity, I think, depending on load conditions, hypertension, myocardial infarction, pharmacotherapy, ACE inhibitors and whatever. All of these things will affect the transcriptome.
Correct. I mean, in terms of metadata, I mean, the regular cardiac risk factors, hypertension, length of hypertension, therapy for hypertension, cholesterol levels, HDL, LDL, inflammatory status, whether that's a CRP or something else where we understand that. So I think that there's a lot of clinical data that would need to be correlated because, again, there is plasticity that's dependent on what we do, what we take, what we eat.
I mean, sometimes a failing heart will recover if it's unloaded for a period of time.
Correct.
So.
Correct.
I would like to turn the question also around. I think the heart can also learn a lot from the atlasing efforts that are going on in the other tissues. And for instance, I mean, we have talked about fibroblasts. It would be crucial to actually compare what makes the cardiac fibroblast different from the fibroblast in the kidney or in the skin. And I mean, you could even perhaps name all of the organ systems that have, to some degree, fibroblasts within them. So I think we also can inform specific organs by putting the data together and look across the different organs, and the same you could do with macrophages and many other cell types that are not specific to the heart.
Correct. Actually, that's a really interesting point. And in the same way that endothelial cells may provide something that allows us to look at different places in the body, fibroblasts may allow us to also ask some interesting questions because they're going to be in every tissue.
Mm-hmm. Mm-hmm.
And innate immune cells are going to be in every tissue, but have maybe different functions.
Mm-hmm. Mm-hmm. Yeah. All right. Good. Okay. Well, I guess we'll go on our break. Thanks to everyone for coming. We have a wrap-up session where we'll try to make some sense of this session and sort of summarize the points. But I'm happy to have met all of you, and enjoy the rest of your conference.